Welcome back to MHC Molecule Series. This is episode 3. I am Dr. Kalpna Rakholiya and today we are going to discuss about structure of MHC molecule. So this is the structure of MHC molecule class 1 and MHC molecule class 2. When you can see this in a MHC structure, there is a four main part are there. Peptide binding group that is close at the both end, while external domain that is extracellular that are present outside of the cell membrane, external domain that contain alpha 1, alpha 2, and alpha 3 and beta 2 microglobulin. While in case of this is the transmembrane segment, this anchor alpha 3 part anchor into the cytoplasmic membrane. That's why these are called as a transmembrane segment. And their tail, this is the short tail that is a anchor into the cytoplasma. That's why it's called as the cytoplasmic tail. Here this part is a co-receptor binding site. Here this is the MHC class 1 structure. So this is it bind a CD8 receptor of the T cytotoxic cell. So here you can see here this is a peptide binding group in which there is a that carry total 8 to 10 amino acid represent to the T cytotoxic cell. While in case of this is the structure of MHC class 2 molecule, here you can see here this is the peptide binding group open at the both end. Here you can see here both hand are open, and the size of the peptide that is the 13 and 18, 30 to 18 amino acid. Sometimes the longer, so we can compare that. MHC class 1 that will carry the small amino acid like peptide like 8 to 10 amino acid, while in case of MHC class 2 which carry the 13 to 18 amino acid. So this is the peptide binding group. This part is the external domain, extracellular structure in which alpha 1 domain, alpha 2 domain, beta 1 and beta 2 domain. This is the transmembrane segment. This is the here main two comparison is that between MHC class 1 and class 2. Here transmembrane segment only present of the alpha 3. Here transmembrane segment uh, both chain will anchor the transmembrane segment as well as the cytoplasmic tail. So beta 2 and alpha 2 both are having a transmembrane segment. Here we can see that CD4 part of the T helper cell will bind here this segment. So let's discuss about how you can draw in the examination this figure. So I have prepared. So this is here you can see that this is MHC class 1 structure. This is MHC class 2 structure. This is peptide binding site and this part is a carbohydrate. We already told that in MHC molecule that is made up of glycoprotein. It means major part is a protein and small part is a carbohydrate. So here you can see that this is the peptide binding site. This is the alpha 1, alpha 2, beta 2 microglobulin. Each part is bind with the disulfide bond alpha 2, alpha 3, and this is the anchor into the cytoplasmic. This is the tail of the cytoplasmic cytoplasma, and this is cytoplasmic membrane, and we can say that transmembrane segment. This is the plasma membrane. So for MHC class 1 that will present the antigen to the two cytotoxic cell CD8 having receptor, and that found in all nucleated cell. And here you can see that here a peptide binding site between alpha 1 and alpha 2. Here you can see that this is a peptide binding site open binding group, and that is a between alpha 1 and beta 1. Here we already know okay, that all nucleotide cell contain MHC1 molecule, but in case of MHC class 2 which only found on antigen presenting cell. Here a peptide binding between alpha 1 and beta 2. Here main component are 2 alpha and 2 beta, beta 1 and beta 2. Here in case alpha 1 domain 3 alpha, alpha 1, alpha 2 and alpha 3, and one is the beta 2 microglobulin domain. So this is the structure, easily you can remember. So let's compare quickly summary of this lecture. A peptide binding domain between alpha 1 and alpha 2. Here alpha 1 and beta 1. Peptide length, it is a 8 to 10 amino acid. Here in case of MSC class 2, that is 13 to 18 amino acid. Groove is closed at both hand, in wide open in both hand. Peptide anchor residue, two major anchor residue are present. Here multiple anchor residues are present. Antigen source, endogenous source, and here is a exogenous source. So pathogen are endogenous like virus particle, tumor, antigen, like that. While in case of exogenous antigen, bacteria, toxin, it present to T cytotoxic cell containing CD8 positive receptor. Here you can see that it represent T helper cell having the CD4 positive receptor. It distributed all nucleated cell while it only present on antigen presenting cell. So, for example, the macrophage, dendritic cell, and B cell. These are the MCQ that may be useful in NET exam, GSET, GATE, ASET, MSc interest level, and so on. So for a competitive exam related to life science, so you can solve this MCQ. 10 MCQ is here, and give me answer into the comment box, right? Thank you very much for your attention.